In this pre-lab lecture, we'll be looking at our second spectroscopy in organic chemistry, and the most powerful one, of course, the nuclear magnetic resonance spectroscopy. This spectroscopic technique is very powerful as it can determine structure of almost all organic compounds. In organic chemistry, we use NMR spectroscopy to identify hydrocarbon skeleton in any functional groups. Experimentally, it only uses sample in micrograms to give us detailed information about the structure of the hydrocarbon in that organic compound. Because it's not destructive, NMR is used even to measure the structure of biological molecules, for instance DNA. Even NMR is employed in analysis of dangerous materials for this purpose. So large information such as the number of protons in a molecule and their relative positions can be obtained from the NMR analysis, and NMR operates based on a certain nuclei of certain atoms, and those include carbon 13, fluorine 19, and phosphorus 31. In proton NMR, for instance, we can determine qualitative and quantitative characteristics of different hydrogen within a molecule. For carbon NMR, similarly, it's possible to identify the carbon skeleton of that organic compound. So the nuclei, such as nuclei of a proton and a carbon, generates magnetic field, and the magnetic field generated by the nucleus can be in plus to half spin or minus to half spin, which means the magnetic field in plus half spin will be opposite in direction from the magnetic field that's generated by negative half spin. So in the absence of magnetic field, these spins, the nuclear spins, will be randomly oriented. However, when we apply an external magnetic field, it causes these spins to align parallel or anti-parallel to the magnetic field. So the frequency absorbed electromagnetic radiation by these spins is proportional to the applied magnetic field as well as to the energy difference between these two spins. Thus, NMR mainly focuses on the change in the direction of the spin orientation, which we also refer to as nuclear magnetic resonance, which happens in response to radio frequency radiation. Because the frequency of absorbed electromagnetic radiation is different for different elements, even for different isotopes within the same element, it will be possible to identify the characteristics of every element, every atom within the structure, within a given organic structure. So the frequency of absorbed electromagnetic radiation for a particular nucleus, such as the carbon and proton, depends mainly on the molecular environment of the nucleus. When we say environment, depending on how the hydrogen and the carbon is attached to a given structure, it will absorb the radio frequency in a different way, and that will give us different peaks in the NMR spectrum. Chemical shifts are resonance frequency of a nucleus relative to a standard in a magnetic field. So protons in different environments have different chemical shifts, which means they have different degrees of shielding, and this shielding is interpreted with respect to the high shielded reference compound, the symmetrical trimethyl and based on that, the structure and the environment of that given hydrogen or carbon is determined. So in NMR, compounds that has less electronegativity difference between in their bonds will be highly shielded and appear to the right side near to the reference compound, the tetramethylsilan, whereas compounds which have high electronegative bond will be deshielded and appear to the left side of the chemical shift reading. So like I said electronegative bonds have I can say polar bonds have bonds are highly deshielded and will have high chemical shifts and appear to the left side in the chemical shift reading, whereas the bonds which have less electronegative bonds or nonpolar bonds are highly shielded and appear to the right side of the chemical shift. So if you take a look, the carbon-carbon bond is less electronegative compared to the carbon-nitrogen bond. That's why the carbon-carbon bond have less than one chemical shift, whereas the carbon nitrogen whereas the carbon it was nothing, the carbon nitrogen will have high electronegativity, so it appears to the right side of the to the left side of the chemical shift. This electronegativity difference is a cumulative shift, which means the more electronegative elements we have in that organic structure, the more deshielded it will be and the higher the chemical shift it will have. So for methyl chloride, the chemical shift will be 3.1 parts per million, whereas for trichloromethyl, the chemical shift will be highly deshielded and appear to the left side above 7. The chemical shift is also so basically the electronegativity, if you think of electronegativity, it is simply movement of electrons. There is electron density in one of the bonding atoms. Similarly, here also protons attached to sp2 hybridized carbon are less deshielded than those of the sp3 carbon atoms, because protons that are attached to sp2 hybridized carbon face magnetic interaction with the delocalized electrons of with the delocalized electrons in the structure, and they will be more deshielded. So you can see that the sigma bond, the ethane with only sigma bonds has no any electron density that affect the electronic and magnetic properties of the hydrogens, and it will be very shielded, whereas the hydrogen, the allyl hydrogens will encounter electronic and magnetic pressure from the delocalized electrons in the double bond, and they will be deshielded and will have higher chemical shift. Even if you see, if you compare the alkene double bond and the aromatic double bond, the aromatic double bond, in the aromatic double bond, the pi electrons are delocalized, they're delocalized and resonance stabilized, of course, and because of that, because of this electron movement in the structure affects the electronic and magnetic properties of that hydrogen, they will appear very deshielded in the NMR reading. So in general, what can we obtain from the NMR spectra? The number of signals in the NMR spectra tells us how many different types of hydrogen are there in that molecule, whereas the chemical shift, if they appear to the left side, it means each of the hydrogens are under an environment with high electron density. It could be resonance, it could be inductive effect, whereas if the protons have a chemical shift near to the standard compound, meaning to the right side, which means those protons encounter less electron density or electron pushing or pulling from the neighboring atoms, whereas the relative areas under the relative areas under signals tells us how many hydrogens of each environment are available. So if hydrogens in one carbon have the same environment, then they will have high area, high relative area, and if hydrogens in the same carbon atom have different environment, and they will have relative area compared relative abundance with respect to the neighboring hydrogens. We will describe this very clearly with an example. A splitting pattern is a situation in NMR spectroscopy where a given hydrogen atom, the electronic and magnetic properties of a given hydrogen atom can be affected by other neighboring hydrogen atoms, and we will have a peak with a splitting pattern, or we will have a pattern, we will have a band with splitted peaks. So let's look at these examples. First structure is cyclohexane, and we are talking about proton NMR, and you do know that in cyclohexane we have 6 protons. However, in all these 6 protons have nothing in difference with respect to the overall structure of the cyclohexane. The proton at the top and the proton at the bottom have the same relationship with the neighboring atoms and with all atoms, I would say. So they will have a single peak in NMR spectroscopy. Similarly, if you look for benzene, all the hydrogen atoms in benzene have the same environment. As a result, they will have single peak in the proton NMR. If you go to the right side, for instance, for 2,3 butene, all the methyl groups have the same, because we are talking about protons. The middle carbon atoms that have the double bond has no any reading. They will have no any signal because they do not have proton with them, whereas all the other methyls are equidistant to the double bond and they will have the same reading, and for this structure you will expect only single peak appearing. Whereas if you see at the paramethyl toluene down to the right side, we can see that we have a proton in the methyl, but we have also a proton which is part of the ring, and this tells us that the proton in the methylene are not equidistant with the proton in the ring itself to the double bonds of the ring. That's why we have 2 peaks for this one for the methylene groups and the second for the aromatic hydrogens in the structure. Similarly, you can see at these structures that with respect to so this is a tertibutyl bromide, and the middle carbon has no hydrogen and it doesn't matter, or it will not have any signal in proton NMR. Whereas if you see the 3 methyl groups, they are equidistant with respect to this carbon and with respect to the bromine itself, which means they are chemically equivalent and they will have a single peak in NMR spectrum. You can also look at the other structures. For instance, in ethyl bromide, the methylene hydrogen are very near to bromine compared to methyl hydrogens in the CH3. Therefore, in this structure, in ethyl bromide, we have 2 different environments. If you remember your knowledge about, if you recall the concept about inductive effect reduces as we go from the first carbon attached to electronegative atom far to the left or to the right, and that tells you that the electronegativity effect reduces as the number of carbon atoms increases from the electronegative atom. That you can relate it with the shielding and de-shielding, because the farther carbon atom from the electronegative element here, the more shielded it will be. So simply for ethyl bromide we will have 2 we will have two peaks appearing in the proton NMR. What do you think? How many peaks do you think we will have for proton for propyl bromide? How about for 2-chloropropane? Take your time and try to give your quick guess or your base for this one, and you will have the answer later on in the coming slides. Similarly, these organic structures have different environments with respect to the electronegative atom. If we see the first one, I can see that all the protons are different with respect to the bromine, and they will have different signals. For the first structure, for instance, I will have 4 peaks. In the second structure, you can see that the middle methylene hydrogens are equidistant from both the chlorines, and they will have their own peak, whereas the methylene groups between the methyl and chlorine are not equidistant from both the chlorine atoms in the structure. Therefore, for this one, you will have 2 peaks. And for the structure at the bottom, which is a methylene chloride, orthomethylene chloride, toluene, and looking at the structure, you can see that the protons in the methylene are very close to chlorine, and they will have different chemical shifts compared to the methyl group. In fact, you do not see any chlorine in the methyl group, so certainly these protons will have different chemical shifts. But also you can see that we have hydrogen atoms for the ring hydrogen atoms. These ring hydrogen atoms can be considered to be nearly similar, and they will have almost the same peak. So for this structure, we will have 3 peaks: one from the methyl, second from the methylene, and third from the ring hydrogens. So in general, as we go from primary hydrogens to hydrogens bonded to electronegative atoms, the de-shielding increases. So primary hydrogens, like in methane, for instance, or in ethane, will have chemical shift less than 1 most of the time, whereas hydrogens that are connected to more electronegative atom will be more de-shielded and will have higher chemical shifts in the NMR spectrum. You can also use this table, this chart, to quickly identify where a given hydrogen environment exists. So like I said, the hydrogenous more polar hydrogen bond will be highly de-shielded and appear to the left side, whereas the less polar hydrogen will be highly shielded and appear to the right side. Toluene. So toluene is an organic compound, as you can see, having a benzene ring with a metal attached to it. So clearly you can see that the methylene hydrogens are 3 bonds far from the ring double bond, whereas the ring hydrogens are 2 bonds far from the ring double bond. That tells you the effect of the delocalized electrons in each of these hydrogens. That's why you can see 2 peaks for this for the toluene molecule. So these are the answers for the first slide. This we we described a little bit earlier. So like I said, for benzene and cyclohexane, we have only a single we have same environment for all hydrogens, and we will have single peak in NMR. That's also true for the 2,3 dimethyl butene, because if you see, each of the hydrogens are 2 bonds away from the double bond. Okay, but try also to see here that when we talk about proton NMR, we only refer to the carbon that have hydrogen atoms. In fact, you can also take the carbon that does not have a hydrogen atoms as a reference to estimate the environment of the hydrogen atoms in there. For instance, in the previous example for tert-butyl bromide, you can see that the carbon attached to the bromine has no hydrogens, so it can be used as a reference. So from that carbon atom, the hydrogens are equidistant. So it doesn't matter how many hydrogens we have. If they are equidistant, it means they have the same environment. Yeah, for this paramethyl toluene, the explanation is similar. You just have 2 kinds of protons, proton in the methylene and proton in the ring, and they will have 2 peaks in the NMR spectrum. Yeah, these are the answer for the previous exercises we were talking about, and you can take time to refer them on your own. Similarly, these are the answers to give you a better understanding of the NMR of an organic compound, and it's good to practice this spectroscopic identification with different examples, and that's why many of the spectroscopic lessons include practice exercises, right? So you can practice the exercises first, and you can refer to these answers later. Integration in NMR tells you the relative abundance or the relative ratio of each of the hydrogen types. If you see at the propyl bromide, we have 3 different hydrogens in 3 different environments. In the C environment, you'll have 2 hydrogen. In the B environment, you have 2 hydrogen, and in the A environment, you have 3 hydrogens. So the ratio of the peaks in the spectrum will be appeared with respect to the number of hydrogens in the same environment. So for this one, the ratio of the peak will be 3 to 2. That's also the intensity of the peak with respect to each type of hydrogen atom. In the example below that, you can see that both the left and right methyl groups are under the same environment. Okay, therefore the intensity of the peak will be 6 times higher than the intensity of the peak for B, because B have only one hydrogen atom. So let's see, in the cyclohexane structure we have hydrogens. In fact, all the hydrogens are in the same environment in 3 of these structures, and the area of the peak is simply the sum of all the hydrogens available there. Where, however, if you see for paramethyl toluene, we have hydrogens in 2 different environments. The hydrogens in methyl group are 6, and they have same environment, and the peak for these methyl groups will be 6-fold, whereas the peak for the ring hydrogens will be 4-fold. They'll be like in 6-4 ratio or 2:3. Yeah. So for tertibutyl bromide, where all the hydrogens are the same in the same environment, simply the peak is the sum of the area of the peak is the sum of the hydrogens. Most of the time the integration is very important when you have hydrogens in 2 different environments, as in the case of ethyl bromide. So the methylene hydrogens are 3, like one, how to say this. So the peaks will be 1 hydrogen longer than the peaks in B. So the ratio of the peak of the methyl group is 1 higher, 1 measurement or 1 intensity higher than the ratio of the peak in B. It's just the same thing for the other examples you see below these exercises. Simply the integration, the length of the peak is integrated, or the length of the peak is the ratio of the hydrogen atoms in the same environment within that compound. Same thing here, but try to because this is an introductory course for you, try to look for different kinds of integration in different organic compounds, because sometimes they can also be very tricky. For instance, you can see in the structure that contains the benzene ring, we have 3 kinds of hydrogens, and these 3 kinds of hydrogens appear in 3:4 ratio. So clearly you can see that the peak for the aromatic hydrogens is twice compared to the peak for the methylene hydrogens, whereas the peak for methyl is 1 higher, a little bit higher than the peak for the methylene hydrogens. That's what the integration, the area integration, is all about. Splitting, another very interesting concept in magnetic resonance spectroscopy. Splitting occurs as a result of magnetic interactions between neighboring non-equivalent NMR active nuclei. Remember, you will only talk about splitting if you have neighboring hydrogens, which means the hydrogens are at least three bonds far from one another. I mean, maximum three bonds far from one another, but they can also be one bond far, two bonds far from one another, but they should be maximum within three bonds far between one another. They should be non-equivalent, which means the hydrogens have different resonance frequency depending on the nature of their attachment in the parent compound. Let's explain this with an example. If you see 1,2 dichloroethane, all the hydrogens are equivalent. Four of the hydrogens are equidistant from the carbons and from the chlorines. That's why you will have only single peak. If you shift one of the chlorine atom to one of the carbon atoms, you will find 1,1 dichloroethane. And clearly, the hydrogen to the right have different environment, or it have different electronegativity impact from both of the chlorines compared to the three hydrogens in the methyling group. Therefore, that hydrogen will split the the three neighboring hydrogens into quartet, which means that single hydrogen will magnetically interact with the three hydrogen in the methyling group and split them into four, whereas the three hydrogens can magnetically interact with a single hydrogen and split it into two. That's what a splitting means. Common splitting patterns are doublet, triplet, quartet, and quintet. In advanced organic chemistry courses, you can also find pentet, sextet, and heptet. But most of the time, splitting above the quintet are called complex splitting or multiplates in general. Splitting pattern follows n+1 rule for n number of neighboring protons which are 3 bonds away. In the first example, you see the proton, which is the only proton in the structure, is supposed to be non-coupled, so you will have only singlet. Whereas at the bottom, you can see the blue hydrogen is 3 bonds away from each of the red hydrogen atoms, which means we will have splitting, and that blue hydrogen will split the three hydrogens into 4, because the neighboring hydrogens are 3, and the splitting will appear as 3+1 or n+1, and you'll have a quartet. So you can understand the splitting by simply knowing the number of neighboring atoms and adding 1 to the number of neighboring atoms. If you have no any neighboring atoms, the peak will be singlet. If you have 1 neighboring atoms, and the peak will be split into 2. Okay, Pascal pattern also put them in a mathematical way so the Pascal pattern works as, so if you have two hydrogen atoms, so a single hydrogen atoms two hydrogen atoms will split one of each other into two, and you will have a doublet. So the Pascal pattern also tells you about the intensity of the peak, because if you add any of the above numbers, the sum of them you'll find below that number. For instance, 1 plus 1 is 2, 1 plus 1 is 2, and the intensity, so that will give you an intensity of a triplet in 1:2 and ratio 3. 1 plus 2 is 3, which is triplet, the intensity of triplet, so okay, so one hydrogen, one proton split neighbor with 3 2 1 hydrogens will have a quartet, will be split into quartet in an intensity of 1:3:1 ratio, right? So that's what a Pascal pattern basically is. But you can also use the simple way, like for n neighboring hydrogen or protons, you'll have n plus 1 splitting. Notice that here, if you want to use the Pascal splitting, you should make sure that the hydrogens, the neighboring hydrogens, are equivalent, and specifically for some hydrogens, for instance, the OH usually does not split, and does not is an exceptional for the Pascal splitting pattern. In fact, the OH will have always a singlet that appear in between 1 to 5.5 chemical shifts, depending on the nature of the OH in the overall organic compound. So we talk about splitting pattern only when we have non-equivalent hydrogens. Okay, so for cyclohexane and benzene or 2,3-dimethyl butene, you have equivalent hydrogens, okay, all over the structure, which means there is no way to speak about splitting pattern. So they're just they just appear as a singlet. So for this structure itself, none of the hydrogens 1 2 3 and 4, so the methyl hydrogens and the ring hydrogens are 4 bonds far to one another, right? So they are chemically equivalent. So the hydrogens are chemically non-equivalent, but they are non-neighboring too. So they simply appear as a singlet, and we cannot talk about splitting pattern in these structures. The same way, that's the same for tertibuthyl bromide. But if you see at the ethyl bromide, you can see that the two hydrogens are in different environments, as we have talked before, and then the methyl hydrogens near to the bromine will split the neighboring 3 hydrogens into quartet, whereas the methyl hydrogens in A will split the 2 hydrogens near to the bromine into triplet. in such a way you can you can use this to understand the splitting in uh, propyl bromide or 2-chloro propyl. Okay, yeah, same thing. uh, you just have to, so when you want to do about the splitting, you just have to make sure that the hydrogens are non-equivalent and they are also neighboring, right? They are separated by at least, I mean by maximum by 3 bonds. And to do one from this, let's choose the second structure. This structure is 1,3 butyl dichloropropane. So in this case, the middle methylene groups are one environment, and the terminal methylene groups are second environment. And each of the terminal methylene groups will split the middle methylene groups the same into triplet, whereas the middle uh, hydrogens will split both the right and left protons into quintet. Okay, how many hydrogens do we have from the right and left side? You have 2 hydrogen in B, you have 2 hydrogens in the left B and in the right B. So 2 plus 2 plus 1 will be 5. That's why it becomes n plus 1, then quintet. Cyclohexane, same hydrogens in same environments. The NMR spectrum will look like the one you're looking at the slides, just a singlet. For 2,3 dimethyl 2-butene, again all the hydrogen atoms are in the same environment, and they will appear as a singlet. For benzene, all the hydrogen atoms are in the same environment. Again, you will have a singlet. But notice here that even if they are a singlet, their chemical shift varies depending how different electron density exists in that structure, right? So compared to the previous examples, we see the benzene is highly deshielded to the left because of the aromatic pipe bonds available in the structure. In paraxylene, we have 2 kinds of hydrogen: the hydrogen that belongs to the aromatic ring and the hydrogen in the methyl groups. So we will have two peaks for this, which are all singlets. For 30-butyl bromide, again you have 9 hydrogens under the same environment, and you will have a single peak as high as 9 hydrogens, right? So it's, as you can see, it's very prominent peak appearing in the spectrum. Ethyl bromide have two kinds of protons with respect to the bromine, and it will have 2 prominent peaks. However, these neighboring hydrogens are at 3 bonds far from one another, and we expect a magnetic interaction between these 2 protons. And you can see that the methylene bromine splits the methyl into quartet, where the chemical shift is seen around 3.5, whereas the methyl protons split the methylene proton into triplet, where the chemical shift is seen around 1.6 or 1.7. In one bromopropane, you have 3 environments, and the first carbon atom near to the bromine is the one which will have high de-shielding, and it will appear to the left compared to all of the other carbon atoms. And we have 2 carbon atoms here, and the neighboring 2 atoms will split these 2 carbon atoms in the methylene group into 2+1, and this methylene group near to bromine will appear as a triplet to the left side in the spectrum. And you do see that the B carbon here has complex splitting. Why? Because these protons can interact with the 2 hydrogens in the right and with the 2 hydrogens in the left. So 2 plus 2, 2 plus 3 is 5, 5 plus 1 is 6, then you will have a sextet. Okay, that's why splitting above 5, above the quintet, are normally above the quintet, and sometimes above the quartet as regarded as multiplets or complex splitting. C protons, the 3 protons in C interact with the 2 hydrogens in B. That's why they split them into 2+1 and appear as a triplet in the spectrum. Isopropyl bromide, you have 2 protons in 2 different environments. You have 9 protons, of course, 7 protons in 2 different environments, and the middle carbon atom with 1 hydrogen will have a septate or a complex splitting that splits the 6 hydrogens into 7, and the 3 neighboring hydrogens from left and right side will split the single hydrogen in the middle into doublet. Two bromobutane has again like 9 protons in 4 different environments, and because 2 of these protons are so if you see the carbon bonded to the bromine, the proton in that carbon is coupled by 5 hydrogens, and it splits them into sextet, and that's the most de-shielded proton you will have appearing in between 4 and 5 in the chemical shift reading. The A protons, which means the methyl groups, are the less de-shielded protons that appear as a triplet to the left side or to the right side of the spectrum. So nothing is so unique here. Simply we are trying to see the splitting in different structures and different conditions, right? So the basic thing is take your reference carbon, the carbon bonded to any heteroatom, preferably electronegative atom, and count or fix the number, the hydrogen atom there, and try to interpret the splitting of the other hydrogens with respect to that hydrogen and with respect to the successive hydrogens. Orthomethyl benzyl chloride, we have seen this already before, and it have 8 hydrogens, 9 hydrogens in 3 different environments, and for that matter we'll have 3 peaks, right? However, what's interesting here is you see that even if we have hydrogens in 3 different environments, none of them are coupled to one another. Okay, that none of them are neighboring groups. So if you see the carbons attached with the carbons in the benzene ring, the 2 carbons attached with branches in the benzene ring have no hydrogens and they do not have any reading, whereas the hydrogens in the methylene chloride are 1, 2, 3, 4 bonds far away from the hydrogens in the methylene group. That's why we do not have any problem of splitting here, and all the hydrogen environments appear as a singlet. In ethanol, the hydroxyl hydrogen is unique, and it simply appears, it doesn't couple with any of the hydrogens and appears as singlet, whereas the methylene hydrogens will split the 3 hydrogens into quartet and appear at higher chemical shifts because of their closeness to the oxygen, whereas the methyl hydrogens in the terminal carbon split the 2 hydrogens into triplets and appear in the right side of the spectrum. Here is an exercise. It's not a big, it's not such a difficult exercise, but try how many peaks can you find from this ethyl benzene? Of course, 3 peaks because it's already displayed there. But how do you relate the quartet and the triplet and the singlet splitting here? What do you think the splitting of the protons in the benzene ring and the protons in the methyl and the protons in the methylene will be linked to each of these peaks? This example can give you a clue to the above exercise. In fact, they have the same NMR spectrum with the previous exercise, and that's because the ethyl in the right side and the ethyl in the left side, they are under same environment, so they will appear in one peak, right? So as for that exercise, this para-diethyl benzene, similar to diethyl benzene, will have 3 peaks, and the proton the protons in the benzene ring are the ones uh, with highest electronegativity influence, and they appear highly deshielded to the left side in the spectrum, and they are singlet, whereas protons in the methylene group are quartet because of the three neighboring hydrogens in the methylene group, and they appear next to them a little bit far to the right in the spectrum. And the terminal methylene groups split the methylene groups, the two hydrogen groups in B, and appear as triplet far to the right, around 1.5 or 1.3 or 1.4 chemical shift. The same thing here, to bromo to methyl butane, you'll have two peaks for this structure, and each peak will be splitted because you can see that we have neighboring hydrogens in each of the carbon atoms. So for instance, for the first terminal methyl group, it's surrounded by or neighbored by two hydrogen atoms, and it splits them into triplets, and you shall see a triplet at around 1 in the spectrum. So likewise, we have three. Okay. The interesting thing in this structure is, if you see the methyl group is bonded to the carbon which is bonded to bromine, we have 6 hydrogens. However, these 6 hydrogens are not neighbored with any of the other hydrogen atoms. That's why all these 6 hydrogens appear as a singlet, right? And you can see that B is a singlet and C is also a singlet, because C so C is a quartet and B is a singlet, because these splitting are very close to each other, and their peaks appear overlapping in the spectrum. But the important thing is, we can see that we have 3 different environments which we supposed to have 3 different peaks. However, because of their close impact to the electronegativity from the bromine, 2 of the peaks are overlapped, and literally we will have 2 peaks. An exercise: how many peaks can you see in the di and propyl ether? And the answer is already given. You will have 3 peaks. But how do you relate the splitting of this dipropyl ether with respect to the spectrum? Which of the carbons gives you a triplet appearing to the left side, and which of the carbons gives you a triplet appearing to the right side? 1-propanol. Again, when you see an alcohol, you will have a singlet, a surely singlet for the OH hydrogen, for the hydroxy hydrogen, but you can work out for the other protons. For the first carbon atoms near to oxygen, they are surrounding with two hydrogens from the next carbon atom, and they will read as triplets, and they appear at a chemical shift 3.5. The middle carbon atoms, if you see, as labeled by B, are neighbored by 3 hydrogen atoms from the left and 2 hydrogen atoms from the right. So 2 plus 3 is 5. 5 plus 1 by using the n plus 1 rule will be 6. Then they will split into 6 states, and you will see them appearing at around 1.5 in the chemical shifts spectrum of 1.5. C is a singlet, like I told you, because it's always hydrogen and it doesn't couple with any of the protons. Whereas in D, again, do not decide by counting the hydrogen of the hydroxyl group even in the D. D is coupled only with the B hydrogens. Okay, with the B hydrogens. How many hydrogens do we have in B? We have only 2 hydrogens. Therefore, the coupling will appear as triplets, and these triplets are seen in 1. Actually, the reading, so the A methyls are also triplets, D are also triplets, okay, but D and A have different electronegativity influence. So the triplet for D is the one that have higher chemical shift, and the triplet for A is the one that has lesser chemical shift. NMR spectrum of benzaldehyde. In benzaldehyde, the carbonyl carbon is a very high electron withdrawing substance for the benzene, and it also affects the magnetic and electronic properties of the hydrogens in benzene ring. So unlike that of alkyl substituents, the aldehyde substituents makes a significant difference in the aromatic protons. So for benzaldehyde, the benzene protons will appear in 3 signals. So for benzaldehyde, we have a singlet for the aldehyde hydrogen, and we have doublet of doublets for the aromatic hydrogens. So because the doublets of the doublets are appear nearly overlapped, but we expect kind of to have 1, 2, 3, 4 peaks, and you can see the first singlet appearing at around 9 point something is for the aldehyde hydrogen, whereas you can see also a peak at around 7.8, peak around 7.7 or something, a peak at 7.6. So I think you can clearly see 4 peaks, right? 4 peaks. So that's the spectrum for benzaldehyde. But the 3 peaks close to each other, you can also see splitting, because these hydrogen atom, the aromatic hydrogen atoms, are in a 3 bonds far from, within the range of 3 bonds far from one another, and they can split into triplets and doublets of doublets. For benzoic acid, again, similar to that of benzaldehyde, you will have 3 protons, 4 environments of protons, one for the carboxylic hydroxyl group, and the 3 other, as you can see, labeled as blue, green, and red for the aromatic hydrogen atoms. But I don't know why the spectrum only appears for only aromatic hydrogens. There has to be another spectrum. I think the purpose here is to show you that you can elucidate structure of organic compound from this software, which you can find at this online site, www.stolaf.edu. and if you can enter the number of compound and the kind of nuclei you want to investigate at different hertzes, you will be able to get the spectrum. Isopentyl acetate is an organic compound with an interesting spectra, because this compound can also appear with spectra that are not expected. And as you can see, the methylene of the methylene next to the carbonyl carbon is surrounded with is neighbored with no any protons and appear as a singlet. And in general, we expect to have 1, 2, 3, 4, 5 signals. However, if you see, you have 1, 2, 3, 4, 5, and 6 signals. So the signal around, in fact, 7 signals, because you have a signal at about 3.7 and 2.1 something. Those are impurities. Actually, this is what makes research very difficult, because when you do research, you're not going to clearly find the spectrum we are talking about. Even for benzaldehyde itself, you might not find 4 clear peaks like that, because as far as you are doing a research, you may have impurities. Okay, it's impossible to be very isolated from any impurities. The pKers we use can be clean, but not as clean as to synthesize a pure organic compound. Anyway, you're going to have impurities. So in research, it will not be easy as such to just elucidate structure of organic compound. And there are also many other parameters that we do apply, and this slide is to show you that you should be cautious in for such spectrum readings, right? Along with proton NMR, carbon NMR is also very common. In fact, both of them are necessary. Most of the time we use both carbon and proton NMR to clearly identify the hydrocarbon skeleton. Like that of NMR, like that of proton NMR, the signals for carbon NMR tells you how many different types of carbons will there be, and the splitting, unlike to that of proton NMR, is not for carbon, but it's for hydrogen. And chemical shifts differ based on the again the electron density around the carbon atom, and the more the carbon atom near to the electron density, the more de-shielded it will appear in the NMR spectrum. And simply the chemical shift is a measure of the environment of that carbon atom, how that carbon is bonded to the neighboring carbon atoms. For instance, take a look at this 2-bromobutane. The 2-bromobutane have 4 carbon atoms. With respect to bromine, each of these 4 carbon atoms are different. That's why you will have 4 peaks, because 4 of the carbon atoms are different. The first attachment, the first carbon atom, as you can see, level D, will have high electronegativity influence, whereas the methylene and the methyl group will have similar electronegativity influence, but because that carbon atom, the carbon atom in C, is further bonded to another methyl group, it will appear as with a different radio with a different radio frequency in the NMR machine, and will have a separate peak with the carbon appear seen in B.